So today I will be talking about another method of delivering trehalose into the body, but I will also be talking about a method of distributing trehalose throughout cells. So let's go ahead and get right into this. This delivery method involves these synthetic structures called liposomes. They're basically these droplet-like fatty structures with a lipid bilayer, which we see all the time in cells. It's useful for getting in and out of cells, in and out of the blood-brain barrier, or just getting stuff in and out of the liposome itself. So the uses of liposomes, well, it can be used to transport things such as production systems and circuits, which we kind of need in a cell, which we want to be producing trehalose and not just delivering trehalose to, which I will get into more detail about later. So the first big plus about liposomes is that it can get through the blood-brain barrier through endocytosis, automatically a plus. And I guess today I'll be talking about trehalose and liposomes delivering trehalose into the blood-brain barrier, or through the blood-brain barrier, and dealing with dementia-related diseases. I know in recent trehalose videos that's exactly what I've talked about, but trehalose is just so correlated with these dementia-related diseases where there are alpha-synuclein buildups in Parkinson's or beta amyloid buildups in Alzheimer's. Is just very useful in that area. Anyway, so I already talked about the blood-brain barrier. Also, liposomes can easily get in and out of cells, just send it in with an endosome. So that's your second plus right there, right off the bat. Alright, so in all of my other videos about trehalose, this is as far as we had gotten. So we delivered the trehalose. You could use a device such as CRISPR-Cas9 to edit your cell, in this case neuron's genome, and produce trehalose, which should enhance the autophagocytosis and untangle all your tangles or your plaque buildups. But the problem is that putting in a strand of DNA into your genome for every single neuron, or just in a group of neurons in a region of the brain, it's very difficult. So a simpler way to do this would be to insert some liposomes that have production systems inside of them. So you have your liposomes L1, L2, and so on. You insert these, and so the thing is that each of these has a certain function because they're not very large. So instead of having one big system, such as one big CRISPR-Cas9 device, you'd have a bunch of tiny parallel circuits really running different functions, which would in the end produce a product that would be trehalose. And you would need some way to activate this, which would be the detection of the plaque if it's outside of the neuron, or if it's inside of the neuron, the neurofibrillary tangles or tau tangles that appear within neurons during Alzheimer's. So going into the specifics of how these liposomes work together, so liposomes can be held together by these proteins called snares, which are not just simple circles. They're actually made of 4 alpha helices, but I won't go into that. So let's say L1 is liposome 1, L2 is liposome 2, and so on till the nth liposome. Each liposome has a function, and all of these liposomes held together by snares, they don't work in order like an assembly line. They actually work in parallel to each other. So let's call this first chain of liposomes working in parallel to each other chain 1, and chain 2, and so on to the nth chain. So obviously you need to figure out functions for each of these liposomes, so your end product ends up being the trehalose that you need. So some possible functions would be creating proteins. Your first liposome's function would need to be getting the would be some sort of activation stimulus in your last liposome, or your last liposome chain's overall output would need to be some sort of trehalose product. And so another possibility for a function could be uh, sending some sort of electrical signal to other neurons. So there's an endless number of possibilities. I am still working out the functions for the liposomes needed to create trehalose. So hopefully by the next research log I'll have that. But for now, this is the basic procedure of how this would be determined. So thank you for watching. Also, I am sick, so that's why my voice is all weird. But yeah, I'll see you next time.